my name is Zhaoqi Zhang. I'll introduce the paper submitted to iMeta. The title is Microbiota-Humanization Drives Human-like Metabolic and Immune Transcriptomic Shifts in Pigs. Pigs are considered highly promising organ donors for clinical xenotransplantation due to the physiological and anatomical similarity to humans. This work offers a foundational step toward the development of gut microbiota-humanized pig models for human biomedical research, and potentially, toward the generation of optimized porcine donor clinical xenotransplantation following further validation of functional benefits. This study successfully established gut microbiota-humanized pigs, achieved human-like remodeling of the serum metabolome, and uncovered transcriptional alterations in immune cell subsets. These findings advance our understanding of how human gastrointestinal microorganisms affect host metabolism and the immune function in a porcine model. At the phylum level, significant differences were observed between humans and pigs in Campylobacterota and Spirochaetota. Additionally, the relative abundances of Bacillota and Bacteroidota differed significantly between control and GMH pigs. Metagenomic profiling revealed the enrichment of Bacilli and Bacteroidia in humanized pigs, which are also implicated in microbiota-humanized mouse models. We identified 136 metabolites that were significantly upregulated in humanized pigs relative to controls, resembling human profiles, and 79 metabolites that were downregulated toward human-like levels. To compare the transcriptome profiles of immune cells across species at single-cell resolution, we performed scRNA-seq on PBMCs from humans, control pigs, and GMH pigs. PBMCs were isolated from 2 healthy human donors, 2 control pigs, 27 weeks old, and 2 GMH pigs, 37 weeks old. Single-cell libraries were prepared and sequenced using the 10x genomics platform. We resolved 31 transcriptionally distinct immune cell populations. Notably, GMH pigs displayed a cell composition closer to that of humans, especially in terms of T cells and CD14-positive monocytes, suggesting microbiota-driven regulation of immune composition. T cells are the main component of PBMCs. To resolve T cell heterogeneity across species, we extracted T cells from the integrated scRNA-seq dataset for subpopulation clustering. To further explore functional changes, we performed KEGG pathway enrichment analysis of genes expressed in GMH porcine T cells in a pattern consistent with human cells. In CD4-naive T cells and CD8-naive T cells, upregulated genes were mainly enriched in biosynthetic and metabolic pathways, including spliceosome assembly, RNA degradation, oxidative phosphorylation, ribosome biogenesis, and thermogenesis. In contrast, pathways related to adherence junctions, Th17 cell differentiation, and leukocyte migration were downregulated in CD8-naive T cells. Similarly, upregulated genes in activated CD4 and CD8 T cells were enriched in translational and metabolic processes, such as ribosomes, oxidative phosphorylation, RNA transport, thermogenesis, and protein processing in the endoplasmic reticulum. GNLY-positive CD8 T cells also showed enrichment in ribosome and ER protein processing pathways. To investigate the composition and transcriptional profiles of circulating myeloid immune cells across humans, pigs, and microbiota-humanized pigs, we extracted and integrated PMBC-derived myeloid cells from all 3 groups. Unlike T cells and monocytes, the overall proportion of B cells in PBMCs remained relatively stable across GMH pigs, control pigs, and humans, with no significant difference observed. Among these subsets, CD24 plus B cells exhibited the most notable compositional shift. CD4 is a well-characterized marker of immature B cells and plays a role in B cell maturation and signaling. Studies have shown that CD24 plus B cells in pigs may be involved in genes regulating BCR signaling and cell proliferation regulation. In this article, we successfully established a microbiota-humanized pig model by transplanting human fecal microbiota into antibiotic-treated pigs. This intervention led to substantial remodeling of the host microbial community and serum metabolome, with metabolite profiles shifting toward a human-like state.